Bismillah Rahman Rahim. Dear students, Assalamu alaikum. I am Waqar Ahmad and this is the lecture number 2 on the ribosomes. Today we will study about the chemical composition and the ultrastructure of ribosomes. The ribosomes, it consists of proteins and ribosomal RNA. So there are two types of ribosomes as we have studied, the 70s ribosome and the 80s ribosome. So the chemical nature of these two ribosomes according to the composition is different. For example, the 70s ribosome, it contains more RNA and less proteins. In E. coli, the ribosomal RNA contents are 63% and the proteins are the 37%. Now the 80s ribosomes, it contain more proteins and less RNA. The RNA contents are 40% in yeast. It has been studied in yeast and the proteins are the 60%. Now the ribosomal RNA, we have studied the different types that in 70S it contains majorly it contains the two subunits. These two subunits are the 50S and the 30S. The 50S further it contains the 23S ribosomal RNA and 5S ribosomal RNA, while the 30S it contains 16S ribosomal RNA. The molecular weight of these subunits, they are different and it is calculated in Dalton. Now one nucleotide in RNA, one nucleotide of RNA, it corresponds to 330 Daltons. So the 23S ribosomal RNA, it contains 3300 nucleotides. So if it contains 3300 nucleotides, how we can calculate its molecular weight? So 3300 multiplied by 330. So it will be the molecular weight of 23S. The 650 multiplied by 330. So this will be the molecular weight, and A is also the 120 multiplied by 330. So these will be the molecular weight of 70S ribosomal RNA subunits. Now the 80S ribosomes, it contain the 80S ribosome. It contains 4 subunits. It contains 4 subunits. The 4 subunits are 28S, the second one is 18S, the third one is 5S, and the fourth one is 5.8S. Now the molecular weight of these subunits, it is also different. The molecular weight is also different, and the 28S ribosomal RNA. The 28S ribosomal RNA, its molecular weight is 1.6 multiplied by 10 raised to the power 6 Daltons. So it is also calculated in such a way. Now the other is, it is most of the ribosomes, most of the RNA in eukaryotes. The RNA in eukaryotes, it is double stranded. Its RNA 60%, it is double stranded. So how it is double stranded? Actually, the RNA, it is single stranded, but it can form double strand also. So how it form double strand? So it formed the double strand by hairpin loops. The double strands formed by hairpin loops. By hairpin loops. For example, these are the hairpin loops. So these are the hairpin loops. So how the hairpin loops formed? The hairpin loops formed when there are present the complementary bases. These bases will be complementary to each other. These bases will be complementary to each other. The 28S ribosomal RNA, it have nitrogen bases in pairs. So these are the pair bases. So the nitrogen bases are present. The nitrogen bases are the adenine, the guanine, the cytosine. But in RNA there will be uracil instead of thymine. The 28S ribosomal RNA, the molecular weight is 1.6 multiplied by 10 raised to the power 6 The molecular weight of 18S ribosomal RNA, 18S ribosomal RNA. Its molecular weight is 0.6 multiplied by 10 raised to the power 6 Daltons. So this is the molecular weight of 18S ribosomal RNA. Now the other is the 18S and 28S ribosomal RNA. It also contains methyl groups. The 18S and 28S ribosomal RNA, its characteristic feature is that it contains methyl groups. Now these methyl groups, it help in three-dimensional structure of ribosomes. Three-dimensional structure of ribosomes. So this is the 28S and 18S ribosomal RNA. Now the other is the 5S ribosomal RNA. The 5S ribosomal RNA, it contains 120 nucleotides. So molecular weight will be 120 multiplied by 330 is equal to its molecular weight. The 5.8 is, it is assumed that it is associated mostly with 28S ribosomal RNA. So these are the subunits of the ribosomal RNA, the 28S, the 5S, the 5.8S and 18S. Now the ribosomal proteins. The ribosomal proteins. The ribosomal proteins, the contents of this protein, it is also different in prokaryotes and eukaryotes. In prokaryotes and in eukaryotes, it is different. For example, in prokaryotes, as we have said, that it contains 54 proteins. The 54 proteins, further, they are present in the larger subunit and in the smaller subunit. The larger subunit, it contains 34 or about 33 or 34, and it contains 20 or 21 proteins. The larger subunit, these proteins are represented by L, for example L1, L2, L3 and so on. And the smaller, they are represented by S1, S2, S3 and so on. Further, the proteins of prokaryotes, the 70S ribosomal RNA, it has studied further by centrifugation methods. So when it was by centrifugation, when the 70S it was centrifuged, and also the 70S it was treated with 5 mole cesium chloride, centrifuged and treated with 5 m cesium chloride. So these 70S ribosome, it contain further, its protein are divided into two types. The two types of proteins are that it contain the core proteins and it contain split proteins. It contain the core proteins, and the second one is that it contain split proteins. So how the core and the split proteins are present? If this is the 70S ribosome, this is the 70S ribosomal subunits. So these are the two subunits. This is the smaller one and this is the larger one. So when they are separated, the 50s and this one is the 30s. So the 50s, this one is now the 50s and this one is now the 30s. So these two subunits were separated and then the two subunits were also treated with the cesium chloride. So first, the 50s subunit, it gives some split proteins. It gave some split proteins. And it also, the 30s also gave some split proteins. So these are the split proteins. Now further, the 50s, when the split proteins were separated from the 30s and the 50s, so then the 50s, it became the 40s, and the 30s, it became the 23s, and it became the 40s. Then the other types of proteins were also separated. The other types of proteins were also separated. The other proteins were the core proteins. Now these are the core proteins. So these, the red color, are the split proteins and these are the core proteins. And the 40s, it contains the core proteins and further it contains the two subunits, the 23s and the other is the 5s. And the 23s, when the core proteins were separated, so it contained, now the final is the 16s ribosomal RNA. So this is about the prokaryotic ribosomes. The eukaryotic ribosomes, it also contain the large the proteins in the eukaryotes. So the eukaryotes also contain the larger subunit, that is the 60s, and the other one is the 40s. So the 60s and the 40s combinedly, it contains 70 types of proteins. Now the proteins of the larger subunit, it is represented by L, L1, L2, L3 and so on. So 40 types of proteins are present in the larger subunit, 40, and about 30 types are present in the smaller subunit. So they are represented by S, S1, S2 up to S30. So this is about the proteins of the ribosomes. Now the metallic ions. The metallic ions which are present in ribosomes. So the metallic ions are the magnesium ions. It has a major role. The magnesium ions and also the calcium and magnesium ions and the calcium ions. So these divalent ions, it has a major role in the formation or in the binding of the two subunits of the ribosomes when the dimerization occurs. So this is about the protein constituent as well as the ribosomal RNA subunits constituent. Now the ultrastructure. The other is the ultrastructure of ribosomes. The ultrastructure of ribosomes. Now the next topic is the ultrastructure. Ultrastructure. As we study about the structure of ribosomes, so the ribosome, there are two types. The most well studied ribosome is 70s. The most well studied ribosome is 70s ribosome. So there are two models. Two models were presented for the structure of ribosomes. Two models were presented. So the two models of ribosomes. The first one is the stopper, and that is the Whitman's model. The stopper and Whitman's model. This model is also called, this is the symmetrical model. This is also called symmetrical model. The second one, this model is called Lake's model. This is called Lake's model. The Lake's model, it is also called asymmetrical model. Asymmetrical model. The asymmetrical, this model, it was presented in 1981. So this model, it describes the three-dimensional structure of ribosome. The three-dimensional structure of ribosome. The most accepted model is Lake's model. This one is the accepted model. Lake's model. So according to this model, as we have studied also, that there are two subunits, and this model was also studied in prokaryotes in 70s in prokaryotes. So according to this, so there are two subunits, the 30s. So the 30s subunit, it contain three regions. It contain three regions. The 30th subunit contain three regions in such a way. So this is the 30th. The 30th, it contain a head region. This is a head region. This is the cleft. This region is cleft. Cleft is an important region because here the codon and anticodon binds with each other. Codon and anticodon. The codon is present on messenger RNA and anticodon on transfer RNA. So this is the cleft where the codon and anticodon. This one is the platform. This is the platform, and this one is the base. So the three important regions are the base, the platform, and the head. The cleft is the intermediate between the platform and the head. So as we said, the three regions, so you will see the three regions will be the head, the other is the platform, and the third one is base. So these three regions you will see in any article or something else in any book. Now the second one is the 50s subunit. 50s subunit. The 50s subunit it contain also a ridge. It contain a ridge, a central protuberance, and it contain a stalk. So if you draw the structure of the 50s ribosome, so the 50s ribosome, it is such like this. So this is the 50s ribosome. The 50s ribosome, so it contain ridge. This region is called, ridge. This region is called central protuberance, and the other one, this is called stalk. This is called stalk. So these three are the regions of the 50S subunit. The 50S subunit, it contains a site of peptidyl transferase activity. The 50S subunit, it contains a site for peptidyl transferase activity. A site for peptidyl transferase activity. And the 30S subunit. The 30S subunit, it provides binding site for messenger RNA. So it provides a 30S, it provides binding site for messenger RNA. The 30S, it provides site for binding of messenger RNA. Now the structure will also study it further. We will study further it a detail in also in the translation, that how the complete process occurred. So this is about the lecture number second, and this is the end of the lecture number second. And in the lecture number third, we will study about the biogenesis of the 70S ribosome as well as the 80S ribosomes. Stay with me. Thanks.